Sorry, Davina, you're right, I am. That's what we agreed.
You want.
So our next speaker is Bob Lahue. It gives me great pleasure to introduce him. He's the Professor of Molecular Genetics at the School of Natural Sciences at the National University of Ireland in Galway. So Bob's worked on DNA repair and mismatch repair in particular for a long time, and indeed spent some years in Paul Modrich's lab, who won a Nobel Prize in this area. And he moved to Galway and has continued to work on both repeat contractions and their response, repeat expansions and contractions, and their response to changes in mismatch repair genes. And he's going to talk to us about a very interesting story that he's developed very recently, I hope.
Mutations. So here's a brief visual summary of what I'll be telling you. Hi, everyone. Thanks for joining. My name is Bob Lahue from the National University of Ireland at Galway, and I'll be describing research from my lab studying histone deacetylase 3 as a target to block HD expansion mutations. So here's a brief visual summary of what I'll be telling you, that we've discovered a target, an enzyme called histone deacetylase 3, or HDAC3. And this class of enzymes then modifies other target proteins by changing their post-translational modification. HDACs remove acetyl groups from lysine side chains in target proteins. And in this case, what we've discovered is that HDAC3 seems to work on a very important protein called mutas beta. And the reason this is important in this scenario is that mutas beta is known to then drive repeat expansions, the CAG expansions that occur in the HTT gene in Huntington's disease. So in this model, then HDAC3 has to turn on mutas beta, and mutas beta then can go ahead and contribute to expansions. What we've also discovered is a small molecule inhibitor called RGFP966, and this inhibitor targets HDAC3 and blocks that deacetylase activity. And in doing so, it then can block this activation of mutas beta and block expansions. So in lay language, basically, if we think about these expansion mutations as an out of control genetic train, basically an ongoing mutational problem that's out of control. The idea here is if we can slow down that runaway train, we can then slow the deterioration of the brain in HD. At least that's what we hope to do. So what are these expansion mutations exactly? So in this diagram, this upper rectangle is supposed to be the HTT gene, the gene that encodes the Huntington protein in humans. And it's known that this gene has a bunch of CAG repeats at the beginning. So the DNA sequence is simply CAG, 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 repeated some number of times. And in most healthy individuals, it's around 20 or fewer of those repeats. In the middle is the mutant HTT gene. This is what an HD individual would inherit then. And where there's simply too many of those CAG repeats, it's called an expansion. So this is what the individual would inherit for one or of their parents. And this is what causes HD. In addition, then, in those individuals as they grow, then there are ongoing mutations. So especially in the brain of the HD individuals, there's additional CAG repeats get added to that HTT gene. They make it even longer and longer. So this bottom category, this is called the somatic expansion, happens within the body of the individual. And there's now very good genetic evidence that says that somatic expansions help accelerate the age of onset of symptoms in HD and also accelerate the rate of decline. So these, in a genetic term, this runaway train is quite a negative experience then. And if we could do something to control this train at this point, these somatic expansions, it should in principle delay or slow down then both the onset of HD and the rate of progression. At least that's what we hope to do. So why would we focus on these somatic expansions? And the reason is there's been a number of recent genetic genome-wide association studies, GWAS studies, that looked in HD individuals to find out what other genes besides HTT drive the rate or the age of onset and the rate of progression. And a number of these genes then pointed to DNA repair factors that are known then to drive these somatic expansions. So basically, the more active and the more abundant these repair factors are, the sooner the disease onsets and the faster the patient declines. And I should point out our chair of this session, Leslie Jones, has made very important contributions to those genetic analyses. So if we agree then that we focus on that, then which one do we focus on? There's several of these factors that have been identified through these GWAS. Which one do we study? So my lab chose to study this protein called mutas beta. So it's shown on the left, and you can see mutas beta contains two subunits. One in green is called MSH2, and the one in blue is called MSH3. And in fact, the MSH3 gene was one of those genes that came out of those GWAS studies. So we know this protein factor is important in doing something that affects the age of onset and the rate of progression of HD. We also know from mouse studies, many of which were carried out in the lab of our subsequent speaker in the session, Vanessa Wiebeck, that mutas beta is very important for expansions in HD mouse models. So what's been shown is that knockout of either the MSH2 subunit or the MSH3 subunit of mutas beta blocks expansions in those mice. And in particular, I just point out that MSH3 is of particular interest because it's unique to mutas beta. Mutas beta is the only form in which MSH3 functions, and therefore mutas beta is worthy of further attention. So then how then did we go about testing this? And my lab uses primarily cells grown in the laboratory in tissue culture dishes, and they're human cells called SVGA. And the reason these cells are useful is because we've developed a system where we can actually measure expansions in the cells in the culture dish. So it's kind of a relatively rapid way of assessing what's going on with expansions and understanding their mechanism. So here's a little bit of the nitty-gritty of how the assay works, but I'll just show you one bit of it. This validation by PCR, you can see the starting track then has a lower number of CAG repeats, whereas these things that we measure as expansions actually do have expansions. You can see they migrate higher on the gel because there's more CAG repeats. So that's the definition of expansion. So these cells then, SVGA cells, allow us to monitor expansions in the laboratory. So it was very important to prove that those SVGA cells require mutas beta to do this. It was a required step in our validation here. And so Norma Keogh used CRISPR-Cas9 to knock out the MSH3 gene in those cells. So we created a knockout cell line, which does not make MSH3 and therefore has no mutas beta. And Norma also figured out a way to start with those knockout cells and add back DNA encoding any variant we want of MSH3. So we have now three kinds of cells. We have knockouts with no MSH3, we have wild type with normal MSH3, and we have now add backs with whatever variant we want. And here's just an example of some of Norma's data. You can see in this Western blot, the knockout has little or no MSH3 protein and therefore no mutas beta. Here's the middle is the wild type, the normal levels. And on the right is the case we purposely overexpressed MSH3 to make excess mutas beta. And then when Norma measures expansion frequencies, you can see it goes from low to medium to high. So these SVGA cells definitely require mutas beta for expansions. Okay, what else is required? So this was the surprise. This is where we discovered these histone deacetylases. And I should point out there are 18 different kinds of HDACs in a human cell, but in our hands, HDAC3, one of them is HDAC3, is the most crucial. And in fact, I'll show you that in these experiments that HDAC3 is just as important as mutas beta for driving expansions. So the work of Ashley Frizzle showed using, again, our assay for expansions and now knocking down different targets using siRNA. So in the blue, you can see when she ablates the abundance of HDAC3, she blocks most expansions. And that's also true with siRNA. It's also true if we use a chemical inhibitor on the right. The blue then is an inhibitor specific for HDAC3. And in contrast, control experiments targeting a related HDAC called HDAC1 failed to block expansions either using siRNA or a chemical inhibitor for HDAC1. So this is some of the evidence we have that says HDAC3 is important in these cells. And in fact, it's just as important as mu-beta. So do the two proteins talk to each other? Does HDAC3 work with mu-beta somehow to promote expansions? And the work of Anne-Marie Gannon says, yes, that's correct. So again, here looking at siRNA knockdowns and measuring expansions, you can see that in purple then, the HDAC3 knockdown is very potent at blocking expansions as shown earlier. In teal then is knockdown of MSH2, one of the two subunits of mu-beta. And in orange then is a double knockdown, simultaneously ablating both HDAC3 and mu-beta. And you can see there's no further decrease here. And that's consistent with the idea that HDAC3 and mu-beta work in the same pathway to drive expansions. On the right, as we know a little bit more about HDAC3 from other laboratories, HDAC3 itself is part of a larger complex called SMART-NCORE, where HDAC3 provides the deacetylase function, this ability to remove acetyl groups from target proteins. There is a second HDAC which can be part of SMART-NCORE called HDAC5, but it's HDAC3 that provides the majority of the catalytic activity. So how then might these two proteins work together? So this is the work of Greg Williams, and Greg hypothesized that mu-beta is present in two forms. One form is deacetylated, so no acetyl groups on the lysine side chains. And in this model, then we presume that that form of mu-beta has a very high expansion activity. There's lots of expansions if mu-beta looks like this. However, we know that there are HATs, histone acetyl transferases, which can acetylate mu-beta, and there's one in particular we think is important called CBP and P300, and therefore puts acetyl groups onto one or more locations in the mu-beta subunits. So in this acetyl group in this model, then we presume has some sort of low expansion activity. It's reduced for its ability to drive CAG repeat expansions. The role then of HDAC3 then is to remove those acetyl groups and basically reactivate mu-beta. So we have this little equilibrium between modified on the bottom and unmodified, and HDAC3 has to create the unmodified form to drive expansions. So is that really correct? How can we test that? And this is where this inhibitor comes in quite handy, RGFP966, and its chief virtues are shown here on the right. In the top, it's potent at a very low concentrations. In the middle, it's very selective for HDAC3. It was the best one we could find. So even though we cannot completely rule out the possibility there might be some spillover effect to other HDACs by this drug, we do know that HDAC3 is going to be the preferred target. And the third is other people had shown that this molecule readily crosses the blood-brain barrier. So when injected under the skin of the mouse, the 966 shows up in their brain within about 30 minutes. So how can we use this in our cell experiments? So we can now modify our model a little bit and say, if we put in RGFP966 that now inhibits HDAC3, we should now inhibit this deacetylation process and therefore inhibit the ability of the system to drive expansions. So the first prediction is 966 should inhibit expansions. The second one, which is harder to prove, but we did it, is that 966 inhibition should occur via the mu-beta protein. So it's in this system that it's the mu-beta is the key target and not something else. So how do we test those two predictions? Well, the first one's easy. So we Greg simply did expansion assays in the presence of increasing doses of RGFP966. And you can see particularly on the right that we can block about 85% of all expansions at the higher dose of the inhibitor. So that looks promising. What about, how can we then prove that HDAC3 is working on mu-beta? And in this case, we went to the proteomics databases to look and see what residues on MSH3 subunit, the unique subunit within mu-beta, where are these known acetylation sites? And it turns out there were 5 that have been reported, 4 in red from the mouse MSH3, 1 in blue from the human. And those lysine residues that are in color here are the sites where acetylation had been reported. So Greg simply mutated each of those codons then from lysine to arginine. And arginine is important because it cannot be acetylated. It will mimic the deacetylated form. And therefore, this mu-beta then containing those 5 KRs should then always look active for expansions. So 5 replacements, so it's 5 KR. So Greg put this back into these knockout cells and said, what happens? And our prediction is the 5 KR should mimic the deacetylated state. Therefore, the mu-beta should always look turned on, and it should therefore resist the effect of the HDAC3 inhibitor. And that's what Greg saw. So on the left, in the absence of any drug, you can see in the orange again, now we're expressing 5KR at normal levels in a background which otherwise lacks MSH3. You can see we get more expansions than normal. And this is because we think this equilibrium between acetylated and deacetylated now favors deacetylated and therefore active mu-beta. On the right is what happens when Greg adds 966, and you can see there is some reduction in expansion activity. So those 5 sites we modified are not the only relevant sites for expansions. But I do point out that especially in the right-hand case here with the high concentration of the drug, we still retain about 1/2 of all expansions. So this, to me, is very powerful genetic proof that says that these sites in the MSH3 that were mutated have to be at least some of the key residues for activation by HDAC3. So it's a genetic bypass experiment. By changing the lysines to arginines, we can bypass the need for HDAC3 and therefore bypass the effect of the inhibitor. So how do we think that might be happening? So one way we think this occurs is through subcellular localization, that basically acetylation forces mu-beta, or at least some mu-beta, out of the nucleus away from the DNA into the cytoplasm. And so here's some of the evidence that shows this. Greg tagged EGFP with a portion of MSH3, 40 amino acids that contain those 5 sites. And when he does this and looked at the localization in controlled situation, you can see most of the fluorescences in the nucleus, but when he adds 966, a fair amount of the fluorescence seems to leave the nucleus and go into the cytoplasm. And in contrast, when he does that with 5KR, which is now resistant to this inhibitor, remember, most of the fluorescence seems to stay in the nucleus. So here's our model then. In this model, the outer oval is supposed to be the cell, and the inner oval is the nucleus. So in this model then, MSH2/3 form mu-beta in the cytoplasm, and it can freely enter and leave the nucleus, as has been confirmed by other people. What we've added then is to say that in the nucleus, it can be acetylated by the HAT and deacetylated by HDAC3, and that this acetylated form can leave the nucleus, but it has a hard time re-entering. This is why we see more fluorescence in the cytoplasm when the HDAC is inhibited, and it presumably is why expansions go down, because now some of the mu-beta is in the cytoplasm and away from the DNA. Okay, so this is all very nice in cells. Does it matter in a more HD relevant situation? So this is a study we conducted in collaboration with Silvia Ginés and Nuria Suelves at the University of Barcelona. So this is an early intervention using 966 to see, does it have any therapeutic benefit in HD mice? And we're going to do this looking in these mice before the onset of symptoms, and treat for 3 months, and then measure both the molecular and the cognitive outcomes in the mice. And so we had a bunch of outcomes. Here's the one that's relevant for this presentation. This is looking at expansion frequencies in the striatum. So again, the somatic expansions above and beyond what the mouse inherited in terms of CAG repeats. So on the left is the control with no 966. And you can see that in blue, you can see 72% of all the DNA molecules we looked at had expansions. 21% were unchanged, and 7% were slightly shorter. And this is very similar to what other people have seen, lots of ongoing somatic expansions in the striatum of these HD mice. On the right is what's new here. In the presence of 966, you can see we significantly reduced the expansion frequency. And in fact, this drug leads to actually a number of alleles which are slightly shorter. So the overall mutational load has been significantly reduced. We've slowed down the genetic train in the striatum of these mice. And then Silvia and Nuria showed that in terms of neurodegeneration, it was slowed. They showed protected cognitive function. They showed that it helped normalize the transcriptional pattern in the striatum, and that the drug was well tolerated, at least to the extent that we could measure. So quite a successful study then. So where could this go next? So the first thing I'd suggest then is to try the same experiment looking later in the disease progression, so after the onset of symptoms. So does 966 then slow or prevent the deterioration once symptoms have initiated? The second point in the middle would be that there are now more recent HDAC inhibitors with even better brain penetration than 966. In principle, we could use those at lower doses and therefore potentially reduce any toxicity. And the third point is to understand much more in detail then what the safety aspects are of these different HDAC inhibitors in these mice. So with that, I'd like to acknowledge and thank the people in my lab who contributed to the work in the top. In the middle are our collaborators, including Sylvia Gines, and in the bottom, including our funders, including EHDN. So thank you very much. And I thank you for your attention. I'll be glad to answer any questions. Thank you.
Thank you very much. That was a fascinating talk. Really, really interesting. I have a question from Leslie Jones. Huntington itself interacts with the nuclear repressor NCOR in repeat length dependent manner. Do you think that this might also be relevant in the mechanism of MSH3 acetylation also exacerbating expansion?
Yes, so there's a lot of things that are not known in our system, and that's certainly one possibility. Excellent idea, Leslie, and thank you for bringing that up. I should also point out HDAC3 does lots of other things besides just act on mutas beta. It has clear effects on gene expression, which is why the gene expression normalization pattern in the mouse study was important to do. So it's not as, there are other things going on besides what I listed, but that I was just trying to stay focused on that particular aspect.
Yeah. So in terms of off-target effects, is that something then you're working through at the moment, looking at other issues in different systems?
Off target on other repeats. So I guess the question is, do you inadvertently change other repeats as well?
Yeah.
But we haven't done that. It's an interesting idea. I think what you'd have, so something like a sequencing approach like was described in Vincent's talk would be a really worthwhile follow-up study. Yeah, so we haven't looked at it, but it's another very relevant point, especially for safety.
Yeah. So I have another question from Sarah Tomaselli from IRBM. Did you check if other HDAC family members are affected by inhibition with RGFP966?
So no, we did not check this. What we can tell you is, going by the literature, that only the class 2A enzymes are affected by this, and that's including the class 1 enzymes. I'm sorry. HDAC1, 2, 3, and 8, and it's high, far and away. HDAC3 is the preferred target. So again, in principle, we can't control where the drug goes and whether it hits only HDAC3, but that's a really good question. And again, it's one of many things we'd like to know more about. So excellent question. I don't have a great answer. Sorry.
Okay. Thank you for a fascinating talk. I'm going to hand over back to Leslie now.
Thank you.